Hedwig, thanks so much for sitting down with me. Much appreciated. We're here at the meeting of the Upov Working Group for Biochemical and Molecular Techniques, the BMT. Is the Naktuinbouw using those techniques?
Oh yes, we do. That was actually my job when I joined Naktuinbouw 50 years ago already. So I'm quite old now. But so when I joined Naktuinbouw, I started facilitation, a lab facility for to use molecular markers in the first place to support the DOS examination. Mm-hmm. And so that is our first application of molecular markers at Naktuinbouw. A second I will come to in a minute. So what you see in the Upov community is that in this very group, the BMT, has been some models defined in which it is appropriate to use molecular markers. Mm-hmm. And in the first place, that is the model 1, where you can use molecular markers as predictors for traditional characteristics. Yeah. So we do so. In our lab, we consulted the literature and defined some markers for disease resistances in tomato, which are traditional characteristics, which are also obligatory in the DOS testing. Mm-hmm. And to use molecular markers instead of bioassays, we think you can really improve the DOS testing, make it more efficient, more reliable, more cost-effective, and speed up the system as well. Yeah, so there's a lot of advantages, we think. A second application to support DOS is in the management of reference collections. Okay.
That's model 2.
That's model 2, okay, exactly. Yeah. So there are many crops where you have really a lot of varieties in common knowledge. Mm-hmm. And to use molecular markers can be very beneficial in the choice of your comparing varieties to put in your tests in a reliable way. So you don't want to miss any relevant comparing variety, but it's also you don't want to have tests with where you have to put 50 or maybe 100 comparing varieties next to your application. So that's not doable. It's not efficient, and it is far too expensive. Mm-hmm, sure. So we think by using molecular markers, we can reliably sometimes exclude comparing varieties without risking anything that you miss out, and make the DOS testing more efficient and cost effective. So that is actually the applications we use at Naktuinbouw to support the DOS system. Mm-hmm. In addition, we also do some variety identification testing as a service to companies who have any question on variety identity. That can be simple questions like I see a barren plant in my greenhouse or in my field. Mm-hmm. What is it? Or very complicated matters like suspicion of infringements. And then we can also perform research based on genetics, on molecular markers, sometimes combined with also morphology, to provide some evidence that can be used in a court case, for example.
How is that called?
The program is called Variety Tracer.
Okay. Can you tell me a bit more about that?
Of course. So we developed this service for companies that can come consult us to perform genetic analysis in combination with morphology. And for us, it's very interesting because every project, every case is different. So for us, it will never be a routine test. So we always consult with you as a company, your problems, your research questions, and then we try together with you define the experimental setup which is needed to answer your research questions. So that is our first phase. We together formulate the experimental setup in a project plan, let's say. And after that, we try to collect all the material that is needed. And their independence is very important. It's not sufficient that you bring in the suspected material. So this suspected material should be brought to us in an independent way.
Sure,
so sometimes
obviously.
bailiffs are involved, and so every time is different. And then we can offer you several things depending on your question, and also on the crop, which is. And that is, we can perform a side-by-side DOS test, let's say, because, well, our institute has many crop specialists, of course, and we have a lot of experience doing the DOS testing for many crops. We also have collections, reference collections that can be used under code in this kind of cases. And we have the lab facilities, the molecular markers. And depending, again, on your questions and on the crop, which is an investigation, we decide which technology should be used. So sometimes it's enough to work with old-fashioned technologies like AFLP or SSR. But there are also cases, for example, to prove if there's a parental line used in a suspected hybrid, that you want to go and look deeper genetically. And then now we're developing new technologies based on next generation sequencing. So that's also a technological approach. And in the end, we combine all the results, writing a report, answering the research questions we defined before, and that we deliver to you as a company. And you can then enforce your rights.
Very useful application, I would say.
I think so. I think this is quite unique. There are not so many labs that offer this service. And because we have this independent position, I think that is very useful as well for many people. So,
of course, we know that companies have been using molecular tools a lot. In DOS examination offices, it's not so very common yet, I would say. Some are using it quite a lot. Do you see a trend there?
Oh yes, I see a trend. I see it, as you stated, that for breeding, especially in agricultural crops, but also in vegetable crops. Well, you cannot imagine breeding without molecular markers anymore. So it's marker-assisted breeding, and now I think it goes to genomic prediction. So that this evolves very, very rapidly. And as an examination office, we can hardly keep up with the technologies and with the developments in what's happening in the company. What we see also, we also deal at Naktuinbouw with not only with the vegetable companies, which are large international companies, but we also deal with the flowers and the tree companies, tree breeders, and these developments are a little bit behind. We see now there's an awareness of the importance of genomics and of molecular markers in the breeding process, but it's actually just starting. So there you see really three different worlds, I would say. And we are in the middle of it. Then, the trend in DOS examination offices, what I see is that, well, we have this nice Upov system, which is all about harmonization. And you see the fairly new countries that joined Upov more recently and are not so in the tradition of the DOS testing with the traditional morphological and based on phenotype, and they didn't develop large collections of comparing varieties. These new countries, they tend to develop more and more quicker into the direction of DNA marker to assist this process. And what I see, and that is maybe also a risk of the more traditional European countries, which are very comfortable in their traditional way of performing the US tests, that we might get behind if we don't acknowledge the importance of DNA markers and don't invest in this, because of course it's also a matter of investment and in money and attracting people that will do the work. More scientific, molecular biologists, scientific people, bioinformatics. So it's a new world, and you need specialists to do this.
Clearly, yeah. Here at the BMT meeting, we heard this week some presentations with certain non-European countries profiling thousands of varieties within a certain crop,
Absolutely.
constructing massive databases with genetic profiles. So yeah, there is certainly a gap forming, and you're right. Europe may be starting to lag behind. Earlier, you mentioned the costs as a challenge. Are there other challenges?
Yeah, of course, there are other challenges. What I see as a challenge, and then I refer to the traditional system of harmonization, and that took a long time in the Upov world to harmonize the system based on phenotype. What I see now is countries and institutes develop new molecular marker sets, and every country do it on their own way. Does it on their own way. So what I see there as a risk is that there are no clear rules for DNA marker sets, for example, or for how to perform a good molecular analysis, or which thresholds to use. So what I think there is really an international a need for international harmonization based on DNA markers, because you can look at a variety and describe a variety based on phenotype. You can also look at a variety based on genotype. It's just a matter you just look with different eyes or with from a different angle to the same variety, and you need rules. Also international acknowledged rules, how you make this genotypic description of a variety. And I think there's a lack of harmonization yet. So therefore, I'm very happy that we as Naktuinbouw are now coordinating an international project.
Which one?
It's a project for harmonization of a SNP set for tomato.
Okay.
This is financed by CPVO from the Immodus funds, and all the examination offices in Europe entitled for the US testing tomato join us in this project, and in addition also Koreans, the Chinese and Japanese examination offices join us as well. So this is really a great opportunity to agree on a standard set of SNPs, and also on harmonization of this SNP set. If we succeed in this project, I think I'm very happy. This is a first step for generating also a database based on this agreed conditions, and I think this is very important.
Interesting development. If we see all these fast developments over time, do you think that all variety testing will, or perhaps should, go entirely to molecular markers or tools?
Very good question. Very good question. Well, it's very difficult to answer this one. As a molecular biologist, I tend to say, well, maybe. But now I'm a little bit used to the speed in the Upov system, and I think, well, maybe not in my working lifetime. So there are several aspects I think we have to consider in answering this question. First of all, you see what you need then is a combination between phenotype and genotype. Of course, there's now a lot effort is going on in prediction. So probably in due time, I think when we know more, and we have more scientific knowledge, it will be possible, based on a genotype, to predict how a plant will look like.
Yep.
I think that in due time it's feasible, it's doable. But on the other hand, you see also a different development, and that is genome editing. You probably know CRISPR-Cas, and so on. So it's now quite easy to modify the genome and just modify varieties with increased habits or increased characteristics without changing the DNA, or only with a very small change in the DNA. So probably you won't even see it when you just normally genotype. You won't discover this change, but of course the phenotype or the added value is changed. So these two developments, I think, are a little bit contradictory. So
that could then, that could then only be solved with full sequencing. Are we going there?
Oh yes, we're going there. Yeah, but even then, full sequencing has also some pitfalls, because even if you take one plant and you take some leaf sample or some root samples from the same plant, you will find different sequences. So what does it mean? We really have to be very careful what we are looking at and make conclusions out of it. So all the time, I think, and that's why you need specialists. Yeah?
Clearly. Yeah. Excellent. Time will tell.
Time will tell. And probably if we go this direction, we have to change the definition of what a variety is in Upov. Well,
that's a perfect closing of this video. Thank you so much.
Thank you.